Okay, welcome everybody to this webinar about variation data in Ensembl. My name is Erin Haskell and I'm one of the outreach officers here at Ensembl. And today's session is third in a series of 8 courses. So we've already had the Introduction to Ensembl by Astrid and the Introduction to Ensembl Genes and Transcripts by Emily Perry on Tuesday earlier this week. Today we're going to be looking at variation data in Ensembl and using the Ensembl Variant Effect Predictor with me. This will be followed by a short 5-minute break and then a presentation by Astrid again, looking at comparing genes and genomes with Ensembl Comparer. Following this next week, we have 4 sessions, starting with regulatory build and exploring features that regulate genes with Emily. And then it will be me again, looking at data export with Biomart and how you can custom bulk export large amounts of data from Ensembl. And then the following 2 sessions about uploading your own data to Ensembl, so how you can browse Ensembl with your own data added, and also looking at how to download data with the Ensembl REST APIs, which is a new section for this webinar series. So today's session structure is going to follow the others. So similar to before, we have a presentation. This is going to be split into 2 for this section, looking at Ensembl variation data, so the background to variation data in Ensembl, where it comes from and what we do with it, followed by a short presentation on the Ensembl Variant Effect Predictor with a demo in between. So I'll do part 1 of the presentation and then hop out and show you how to view variation data in the browser, and then I'll pop back into the presentation to give you a short introduction to the Variant Effect Predictor, and then we'll have a demo on how to use that. There are exercises available as part of this course, but these are all available on the Train Online site, so they're available for you to do in your own time. And you also have this solutions to these exercises there as well, so you can work through these, and then if you're stuck, then you can please contact us and we can help you get through those exercises. So today we're looking at variation data first off. So what types of variants do we have in Ensembl? Where does this data come from? How do we predict biological consequences for these variants? And common things to watch out for. So there are a few things that often catch people out, and so we're going to arm you with that knowledge so that when you encounter them, you can understand the data. And then for the Variant Effect Predictor, we're going to be looking at what data can we use with the VEP, how do we identify known variants, and how do we predict consequences for novel variants that you have submitted to the VEP tool. So let's start by defining the variant types that we have. These are defined into 2 broad categories. So we have sequence variants, and these are typically smaller alterations, less than 50 base pairs in length. And then we also have structural variants, which are these larger alterations that can cover over 50 base pairs in length and can sometimes span multiple genes. So starting with the first section, sequence variants, most commonly these are single nucleotide polymorphisms, also referred to as single nucleotide variants, so SNPs or SNVs. And this is where we have an allele on the reference genome, and then an alternate allele is cropped up at the same position, and it's just a single change in a single nucleotide. So in this case, a change from an A on the reference to a G on the alternate. We also have small insertions and deletions of a couple of base pairs, or less than 50 base pairs in length, as I said before. So we have an insertion where a novel allele is inserted into the sequence and lengthens the sequence, and a deletion where the original allele at a certain position is deleted and the sequence becomes shorter. We can also have an indel where both of these happen, so we lose the original allele at a certain position but gain new alleles and lengthen the sequence at the same time. So these insertions and deletions can happen at a larger scale, so they're included also in structural variants when they occur over a 50 base pair length. But we also have other classes of structural variants as well. So these longer variants, we have copy number variants, or CNVs. And these are where we have a repetitive sequence in the genome, and we have a section of that that's either repeated again, so we have a gain in this repetitive sequence, or a loss, where that is, a section of that is lost. An inversion is where we have the nucleotide sequence at a certain position, maybe in the 3' to 5' orientation, and then it's inverted, so it's flipped around within the same location. So it's essentially as it would appear on the reverse strand rather than the forward strand, for example. Translocation is where we have a reference section of the genome, so that is then moved to a new location. So this can be either intra-translocation, so within the same chromosome, or inter-translocation, so across a different chromosome. So a section of sequence may move from chromosome 1 to chromosome 2, for example. So now let's take a look at where this data comes from. we have a sort of 4-step process for importing data into Ensembl. So we don't carry out any sequencing ourselves, but rather we import variant data from publicly available repositories. So we start off by importing data, applying some quality control to make sure that it matches our reference data set, for example. And then we add some linked data, and then we perform our own analyses on these. So I'm going to go through this process just now. So starting off with the variant import, we import data, as I said, from publicly available archives and repositories. The 2 main ones are the DGVA archive, which is where we get our structural data from, and then the dbSNP, which is typically where we get our short sequence variation data from. There are a whole wealth of different resources that we have, and depending on the species, they are sometimes specific to that species. So I recommend that you check out this link at the bottom for your species of interest to see where the variation data comes from. So here's just a screenshot from the link shown at the bottom. So for the main Ensembl vertebrate site, ensembl.org, we have 23 species with variation data available. So the most popular species is human, and you can see listed here that we have 655 million short variants or sequence variants available for human. You can see also here that we have in green text next to human plus 325 million, and you can see from the table header that this is what has changed in the most recent release. So as we move to Ensembl 93, we doubled the number of sequence variants that we have available in Ensembl. If you're working with our sister site, Ensembl Genomes, we also have variation data available for species here, for all of the different sites except for bacteria. So it's worth checking the link at the bottom as well if you're working with Ensembl Genomes to see whether your species has variation data available. So moving on now to quality control. So once we've identified the locations and alleles from our variant import, we can then run some quality checks to make sure that they match what we have in our reference assembly, for example. So some of the databases, particularly for human, may have identified variants from the older human genome assembly. And so we map these coordinates to the newer genome assembly so that our data isn't lost. So from GRCH37 to GRCH38. We run checks on alleles to make sure that what is reported as being the reference allele actually matches what we have on our records as being the reference allele in the genome. And we run checks for these IUPAC ambiguity codes, which we'll have a look at in the demo, but these are essentially codes that are single letters that represent a specific change in a base. So a G to a C will have a unique code, and a C to a G will have a different, also unique code. So we'll take a look at those in the sequence view. And we so here we exclude suspect variants, but we sometimes don't include them in default sets, but we do still keep them in the database. So you'll often see flagged variants coming up, and we'll have a look at those in the demo as well. So now looking at linked data, once we have the location and alleles of variants, then we can have a look at things such as phenotype and disease that's associated with them based on citations in the publications. And looking at allele frequency, so particularly from the 1000 Genomes Project, how common is this variant across different populations and different people. And let's just take a look at a couple of those now. So we have linked data, as said, from the 1000 Genomes Project, and this is probably one of the most important early whole genome sequencing projects there are, and the aim of this project was really to understand sort of a baseline level of human genetic variation, so particularly focused on identifying common variants, so ones that are present in at least 1% of the population, and looking at how these are different across the globe. So we'll take a look at this in the demo as well. And more recently, this large increase in human variants that we saw in the previous few slides is mostly due to this import from Nomad, which is the genome aggregation database. And you can see from this graph on the right that the 1000 Genomes Project sequenced about 2,500 individuals in the end, and the Nomad project has looked at over 130,000, or aggregated data from over 130,000 individuals from both genome and exome sequencing projects. So this resource has got such a high sample number that we're able to identify some really rare variants within the population. So this is another useful resource. So after we have all of this external data imported into Ensembl, we can run our own analyses. So we're able to predict variant consequences by looking at these locations and where they overlap with our gene and regulatory feature models. We're able to run algorithms to look at protein function predictions, so whether a change, a variant that results in a change in the protein coding sequence is going to be particularly significant or perhaps not so damaging. We can run linkage disequilibrium data, for which we use the 1,000 genomes data set for, so looking at whether your variants are co-inherited with any other variants. And looking at variant conservation across species with our using our data from our whole genome alignments, which Astrid is going to talk about in the next session. So let's take a look at these variant consequence terms. I'm not expecting you to be able to read all of this data on the slide, but you can find the link at the bottom here. This is just to highlight the different terms that we can apply to a variant. So these are standardized variant consequence terms defined by the Sequence Ontology Consortium. And if you look along the left-hand side of this table, you can see a range of different colors. And typically the red, oranges, and yellows, these sort of danger colors, represent variant consequences that are predicted to be particularly bad or damaging to the function of the gene. And then the greens and blues are perhaps less serious consequences, such as like a synonymous variant, so it's a change within the coding sequence of a gene but doesn't change the actual protein sequence, so we would predict that to not be particularly significant for gene function, for example. And just to show you, this is a sort of schematic of a transcript, which you will have seen from Emily's presentation, and just to highlight all of these different consequence terms within the context of the gene model. So you can see we have regulatory region variants that affect regulatory regions, and also looking at these coding sequence variants, you can see there's a lot of danger colors falling within the coding regions. So let's take a look at these pathogenicity scores now. So I said that if we have a variant, a missense variant, so a variant that causes a change within the protein coding and therefore the amino acid sequence of a gene, we can run these two prediction algorithms, SIFT, which is sorting intolerant from tolerant, and polyphen, polymorphism phenotyping. And these are able to tell us, based on how conserved the amino acid is and what the chemical change in the amino acid is, how significant this variant is going to be for protein function. So you can imagine if we have a variant that codes for a hydrophobic amino acid, whereas normally the gene at this location codes for a hydrophilic amino acid, then this is going to have a significant effect on the way that the protein folds and ultimately perhaps what binds to it as well. So it can be quite significant, and these scores enable us to have a look at that. So we have both of these scores are rated from 0 to 1, but unfortunately they are rated in the opposite direction to each other. So we have the SIFT score values closer to 0 are more deleterious, whereas in polyphen scores closer to 1 are more damaging or deleterious. So we will highlight these scores in Ensembl. We'll also highlight them with these colors, and then also the terms if you click or hover over these colors in case you are colorblind, for example. So also looking at linkage disequilibrium. So this is the defined as the non-random association of alleles at 2 or more loci within a given population, or put more simply, how often two variants or specific sequences are inherited together. So we are able to look at this for groups of variants and across regions, and we will display this data within the variant and location tabs in Ensembl. But also if you have your own variant data, you can run our linkage disequilibrium calculator tool. So you can define a specific genomic region that you're interested in, or supply it with a list of variants, or for areas surrounding a variant of interest, and to have a look and see if there are any other variants that are commonly inherited with your variant of interest, for example. So that sums up the variation process, so this 4-step process to getting variant data into Ensembl and analysing it. And you can find this data across all levels of Ensembl. So we're going to have a look at the website in the demo, but you can also find it, of course, in the Variant Effect Predictor, which is also available as a script, in Biomart, and programmatically looking at both the Perl API and the REST API. So I did say at the beginning that there are some things that often catch people out, so I'm just going to go through a couple of these now. So most notably is these reference and alternate alleles. So genome sequences are created from real biological materials, so it's only natural to assume that this biological material is going to have some rare and possibly private alleles within it. And so you may find a variant, and the variant in this example, we have an A to a G substitution. So on the reference allele, we have an A at a particular location, and the alternate allele is going to be a G. But we can see that the alternate allele is actually far more common in the general population than the reference allele. However, because this is the allele that was on the actual reference genome, we will still report it first. So this is really just to point out that the reference allele is not always the most frequent allele in the general global population. So we have an example of this. So we have the SNP, RS1169305, which is a missense variant. And you can see the alleles reported A/G, so A being the reference allele and then G being the alternate allele. We can see that the ancestral allele at this location is G, so this is conserved in primates, for example. And then the MAF, which stands for minor allele frequency for the A allele, which is the reference allele, is actually only at 1%. So, and looking at the population genetics pie charts from below, we can see that the G allele at this location is actually far more prevalent across the populations. So this is just something to make a note of. If you identify an allele in your sequencing data, it may not always necessarily be a rare allele. And this is very important to point out. In Ensembl, we always report variant alleles on the forward strand, even if they affect a gene that is encoded on the reverse strand. So we will always, by default, report T to G in this example, but when it's in the context of a gene, we will highlight both our T and G and also A and C as the alternate alleles, which we'll take a look at in the browser demo in just a second. dbSNP will report both, depending on the gene location, and PubMed really depends on what the author who's writing the paper decides to write, either in the context of the gene or the reference allele, and often this lacks further information, so they may not even state which one is which is why we need to always run these quality checks when we import variant data to Ensembl. So, let's pop into the demonstration now. So we're going to have a look at how you can find variants within a gene of interest, and we're going to be looking at MCM6. And then we're going to take this out to a broader view to look at how you can find variants within a genomic location of interest, and also how to find out information about a specific variant as well. So I'm just going to pop out with the presentation now and go to the browser. Okay, so here we are on ensembl.org. I've just come to the home page here, and this was explained in some detail in the previous two presentations, so I'm not going to explain what you see here, but rather just use the search box and search for my gene MCM6. Okay, so we will have seen this results before. I'm just going to click directly on the first option, which is for the human gene. So I click on this. You can see that we've come directly to the gene tab. It's highlighted up here. And we have our gene summary here at the top. So this has all been covered before by Emily in the genes and transcript section, so I'm going to go directly to the section on the left here, genetic variation. You can see that we have three options here: variant table, variant image, and structural variants. So while it doesn't say for the variant table, this is only actually going to show us sequence variants, so these shorter variants, less than 50 base pairs in length. The variant image is actually currently retired for human because of this large import of sequence data. It's not particularly useful for human anymore because the image has just become overwhelmed. But note that this is available for other species, so if you're working in mouse or zebrafish, you'll still be able to use this variant image view. And then the structural variants is looking at these structural variants, which are over 50 base pairs in length. So let's start by clicking on variant table and explore this first. So I'm just going to get rid of this filter I've left on. So here we've come to a table, and I'll just walk us through this. So we're starting off by listing the variant IDs. So these RS IDs typically always come from dbSNP. And these are all listed within sort of location order from the lowest base pair to the highest base pair. So you can see that we're starting off, if we look over here at the consequence type, with a 3' UTR variant. So we were looking really at the from the 3' to the 5' end of a gene, looking at all of the variants that fall within that location. If we go back over here to the alleles column, you can see that we have the reference allele listed here, and then in this case a / to show the alternate, which is nothing. So this is a deletion, which you can see in the class column. And we have the global minor allele frequencies. Now we calculate this data based on the 1000 Genomes project. So these first 5 variants do not have this data, whereas the 6th one does. So this is a variant that was identified within the 1000 Genomes project. And if we look across to this evidence column, we can see we have this icon which says 1K, which is representing the 1000 Genomes project. And you can hover over and see what kind of data is available for these different variants here, and also the source of this data. So our recent import was from dbSNP 151, so all of the novel variants that were identified there are all highlighted by the source column here. So now you might want to explore just specific types of consequences or filter this table in any way, and you can do that with these filter options at the top. So for example, if we want to filter by consequence, we can see all of the consequences listed here. Currently they're all turned on. We have quick links at the top to turn them all off. Select certain ones, so ones that affect proteins, so protein truncating variants, and missense ones, or ones that only fall within exons. I'm just going to turn them all off now, so you can see they've all greyed out. And I'm just going to turn on missense variants, which is now turned green. And you can see listed in the brackets the total number of missense variants that we have available, which is over 1,500 in this case. And then when I click apply, we should see my table filter down quickly to look at only missense variants. You can see these are all listed here. And these are still going to be in the same order, so starting at the most 3' exon. But you can see now that we have these 4 columns here populated, which weren't populated when we're looking at the 3' UTR variants. And these are all specifically related to proteins. So we have the amino acids, so what was present originally on the reference, and then what has been substituted with the single nucleotide polymorphism. We have the location of these, and then these all-important SIFT and polyphen scores. So you can see this first one, the scores don't agree. So we have a red 0, which was predicted by SIFT to be particularly deleterious, whereas polyphen is looking at this and thinking that actually it's probably just benign. So they don't always necessarily agree, but they should give you a good idea of whether this is going to be a damaging or not damaging variant. And you can also sort these, so you can filter your table to find the most significantly damaging ones to begin with. So here we can see this one is predicted to be particularly damaging. Okay, so you can apply multiple filters, and they will reduce the table in conjunction with each other. So you can filter any of the columns present in this table at all. So I'm not going to do any more here, but just to highlight that that's a possibility. And now I'm going to click on the structural variants option to look at the structural variants data for this gene. So first of all, we're presented with this genome browser image. So you can see we've separated our structural variants into what we call smaller structural variants and larger structural variants. And these larger ones are going to really span many, many genes. And this is a collapsed view that we'll look at how to expand in the location tab. But we can see all these smaller structural variants, some of them quite large in size, but much smaller compared to other ones. And you can always click on these if you're not sure of what they are. You can click on them to get the class definition. So this one's a copy number variant, as well as some brief information about the variant. And if you scroll down, you can see that we have this section within the legend that defines all these different colors. If we scroll further down, we have a table similar to what we saw on the previous page that lists all of the structural variants starting with their name or identifier. So you can see these NSV or ESV, which is different to the RS IDs that we got from dbSNP. And then all of the classes that we have here are available, as well as their genomic size in base pairs. So you can see some of these are 139 million base pairs in length, so really very large alterations to the reference genome. Okay. So let's take a look at how you can look at sequence variants on this actual sequence. So if you're interested in taking a look at this, we have options here to view the genomic sequence. And if we click on configure this page, we can see how we can filter this down to see some information that we're interested in. So by default, show variants should be on no, and you can change this to yes and show links. And then you can use this option here to filter variants by consequence type. So I've selected missense variant, but you can just choose whichever ones you're interested in, just to simplify the view. And you can even choose which source you're interested in as well. So I'm going to just click and close this now. So we can see from the sequence image that we have our exons highlighted here with this peachy background and red lettering. And you can see that these annotations have now popped up here. So the yellow colour is the sequence ontology are matching to the sequence ontology term missense variant. So you can see we also have these letters here. So these are the IUPAC ambiguity codes. So we have an M here, and if we hover over this, it will tell us that this represents a C to an A. And if we click on it, we can see a bit more information about that identified variant. If you can see here as well, we have one variant that is represented by W, but has also got a grey background. So if we click on this, we can see here that this has got the status of failed status, which indicates that it hasn't passed our quality control during the import. So this is useful to highlight that we have seen that this is potentially a variant at this location, but the data is not quite right. So you can really consider all of the known variants when you're taking a look at variants within your gene. Okay. And these links on the right-hand side are really just corresponding to the linked variants here that are highlighted in order. So you can scroll along, so there's quite a number of these. Okay, let's now take a look at the location tab. If we click on here at the top, we jump to the location tab. Notice that there's also a variant table available here, which looks at the whole region that we're looking at. So whereas when we're in the gene tab, we're only looking at variants that cover from the 3' to the 5' UTR, this variant table is going to be looking at also variants that occur in intergenic regions, for example. So we're on the region in detail page, which is probably the most powerful way to adapt the genome browser image. So if we scroll down to this most detailed view, you can see that we have 3 tracks loaded by default, and these are 1,000 genomes, SNPs and indels, phenotype associated variants, and this structural variants from the 1,000 genomes project as well. So I've already added this structural variants from the DGVA from the configure this page option, but let's see if we can add any more information available as well. So I can click on this cog just to quickly configure this image rather than scrolling up to find the configure this image page. And we come to this pop-up, and if we scroll down, you can see that we have this section called variation and also a section called somatic mutation. So the variation section will only include germline variants, whereas the somatic mutations are going to be including ones that are acquired through cancers, for example. So if we click on these, we can turn on somatic variants. I'm going to put normal on, and then from a variation we have the DGVA already turned on. And if I just click to close this, it reloads. You can see that we have our somatic mutations identified here. You see these typically occur within the coding regions of the gene. And we have this track here. So this is similar to what we saw in the gene tab for structural variants, a collapsed view of this. If we click just on the name here, we have a quick option to change the track style. So currently it's compact. We can change this to expanded and see all of the structural variants that overlap with this gene, both large and small, within this single image. So this is quite similar to what we saw for the structural variation page within the gene tab. But we can zoom out here and look at the larger region and how far these structural variants cover. Okay, that's taking a little bit too long to load, so I'm just going to go up to the top here. So we're going to now take a look at a specific variant that overlaps this gene, but rather than filter down to find it, I'm just going to search directly for it. So using this search box at the top, I'm going to be interested in this variant RS4988235, and I'm just going to search for it directly and go. So this is a unique identifier for a specific variant within a specific species, so we're only given one option in the output, so let's click on that directly. So here we see that we've come to the variant tab now. So previously we were in the gene tab and in the location tab. So first of all, we're listed, we see the RSIG listed at the top, and we see the most severe consequence. So in this case, we're looking at an intron variant. So because there are often multiple transcripts of a single gene, a single variant may overlap many transcripts, and depending on the function of those transcripts may incur a range of different consequences, from protein coding to intron variant to a non-coding transcript variant. So we always list the most severe consequence first, so predominantly looking at missense variants or stop loss, for example. We can see that this variant actually has two alternate alleles, so the reference allele is this G, and then we have both an A and a C have been identified at this region as well. We can see that the ancestral allele is a G as well, so that indicates that this is probably the G is probably the most common variant. And you can see that the minor allele frequency agrees with this, so we have the A, the second, or the first rather, alternate allele is present at about 16% of the population. So we have a number of different identifiers here for the types of data that we have available for this variant, which I explored in the variant table. And you can expand this to see things such as HGS names and synonyms for this variant. But let's take a look first at genes and regulation. So I said before that this variant I know to overlap the gene MCM6, and the gene MCM6 is a reverse stranded gene. So what we can see from this first table is our stable identifier for MCM6 and also the gene name. And this, in brackets here, we have indicated a negative symbol, so we are told that this is a negative stranded gene. And then when we come to look at the alleles, we know from the top of the page that the alternate alleles are A's and C's, and so we have A's and C's listed here from the forward strand. And then in brackets below hand, we have the alternate allele as it would appear within the gene, which is negatively stranded. So if you're still unsure, you can click on this show option, and it will tell you exactly at the bottom. So allele reference forward strand, and then allele transcript strand. So if you ever get confused, just click on that. We also have listed here some gene expression correlations, which come from GTEx, so where this variant has been identified as having a relationship to another gene in terms of its expression. And if we had any regulatory features that this variant overlapped, these would also be listed in a table here. Okay, so now let's take a look at population genetics. Okay, so there should be some pie charts that appear here, but they're currently not loading for me. But these will list just show a pie chart of the two alleles that are common within the 1,000 genomes population, and these are listed by the superpopulations, and then you can expand the subpopulations to see those listed here as well. But if we come to this table, we can actually see this represented here. So these colours would be what would be shown in the pie charts above, and you can see these are listed by superpopulation and then the subpopulations within that, so alphabetically listed. We can see that the alternate allele represented here in the green is highest in Great Britain, for example. So we can see that the frequency of the two alleles here, but then also the genotype frequency, if I come back up to the top. So these are going to show you which of these genotypes are most common, so whether the reference or alternate allele is present within both copies of the chromosome in humans. So we have the homozygous for the reference allele, GG, homozygous for the alternate allele, AA, and then heterozygous, having one copy of each. So you can see how that is distributed across the populations. And if we scroll down a bit further, we can maybe see, here in UK10K, we have one individual who's been identified as having a C allele at that particular location. But you can see that that allele is incredibly rare. Most of the other resources report just the A allele as being the alternate. So let's have a look and see some phenotype data for this variant. So we come to this table to begin with, which shows us the significant associations with particular phenotypes. So body mass index has been reported, hip circumference, and lactase persistence. So we can see also in this column here the associated allele, which allele is reported to have this linked to this phenotype. So we can see the alternate allele here has been linked to all of these different phenotypes. And if we want to, we can expand this and see evidence. So we have a list of the PubMed IDs of papers that have cited this relationship, as well as these will all be listed here in citations. We can click to see linkage to disequilibrium data, but I'm going to skip that for now, and we're just going to take a look at phylogenetic context. Okay, so to begin with, we have the primate alignment selected. This will differ depending on what species you're looking at. You can select other alignments, which Astrid is going to talk about in her presentation, but these different groups. So we have, you can expand this to a wider taxonomic group if you want to see how conserved this allele is at a particular location across a larger taxonomic group. And then we have listed all the locations of the corresponding, across the corresponding species. And you can see here highlighted in the bold text, our reference focus allele here is highlighted in red. And then all of the alleles at that location are conserved across all of these different primates, so it's always the G allele. Okay, so I think we've explored the variant tab and variant data thoroughly now, so I'm going to go back to the presentation, and we're going to learn a bit now about the VEP. Okay, so the VEP stands for Variant Effect Predictor, and essentially does what it says. So it's a tool to predict and annotate the functional consequences of variants. So it enables you to supply it with your own variant input data. It will cross-check with our Ensembl variation database, which is what we've just explored, and it will give you an output to show you what genes, transcripts, or protein sequences are affected by your variants, whether your variants overlap any regulatory regions, if you're working in human and mouse, and that data is available. And when it can find known variants, it can tell you a wealth of data, which is what we've just explored in the demo. So we're really aiming to represent the whole process from transcription to translation and protein function within the VEP. So we're able to tell you whether your variant overlays a regulatory region and so may affect transcription, whether it may have an effect on alternate splicing, so or protein or transcript truncation, an effect on translation, so binding and the amino acid sequence, of course, and then also ultimately looking at how it affects the protein function using our SIFT and polyphen scores, as well as other additional plugins, which we're going to explore as well. So you can use Ensembl with a range of different data input formats. We have our own default method of reporting variants, which is how we store the data in Ensembl. So you can use that as an input. You can also use HGVS notation, which is quite commonly used throughout the literature, and this can specify variants using IDs or genomic locations. So looking at variants at the cDNA level, genomic level, or protein level. Most commonly, if you've carried out your own sequencing variant calling, then you'll have your data output in a VCF, this variant call format. And you can see this is quite similar to the way that we report it in Ensembl, but simply listed by the chromosome positions and the reference and alternate alleles. And you can also supply VEP with a list of variant IDs if you want to do batch analysis on IDs that you've already identified. And what's useful about the VEP is that you can supply it with any of these input formats, but you can also get any of these formats in the output as well. So it's useful if you want to translate your VCF into HGVS notation, for example. And if you want to see more information about these, the link at the bottom will take you to the documentation about all these input formats. So if you've submitted a list of variants and some of them are already known, then we're able to tell you a lot of information about these. So looking at all the databases that we import data from, we can also tell you how common your variant alleles are in different populations, both from the 1000 Genomes project, but also these other projects, including NOMAD. And we can tell you if we know that a variant has been linked to any phenotype, diseases, or clinical significance, as well as any, of course, literature citations as well for those variants. If your variants are unknown, then we're also able to tell you a wealth of information about those. So we can perform consequence predictions using our own Ensembl database, so looking at our gene models and transcript models, but also through RefSeq, and you can even use both an emerged gene set. So if you are commonly using RefSeq, you can still use the Ensembl VEP to annotate your variants. And also the GenCode basic gene set, which Emily described in the previous session, but essentially is a subset of high-quality protein coding transcripts most often. We can also tell you if your variant overlaps regulatory regions based on our regulatory build pipeline, which is what Emily's going to be speaking about next week. And what's useful about this as well is that you can also limit it to regulatory regions that are known to be active within certain cell types. So we have, I think, 79 epigenomes available for human and mouse, and you can limit your output to only show you variants that overlap regulatory regions that are known to be active in lung tissue, for example. And we can also run pathogenicity predictions. We've looked at SIFT and polyphen already, but we also have a range of plugins, which I'll talk about in the next slide, that will enable you to run additional analyses on your variants as well for pathogenicity predictions. So plugins, these are things that essentially we don't by default do for the data that we present on the website or in any of our databases, but we enable you to use them in the VEP, so you can run additional data and get additional output in your script. So, this is available on the web tool and with the script as well. So we'll look at these in the demo. What's amazing about the VEP as well is that you can use it for any species. So you can access it through the web browser REST API or Perl API. As I said in the previous presentation, we have variant data available for 23 species in the main Ensembl site. But you can use the VEP for any of the species in Ensembl, because essentially you can either check for known variants or identify consequences for novel variants, and you're essentially just doing novel variants when you're using the other species in the VEP. And this is true for Ensembl genomes as well, with the exception of, with no exceptions actually, you can use it for any species in Ensembl genomes at all. And in addition, you can actually use it with any species that's not available in Ensembl as well. As long as you have a GTF, which is a gene annotation file, and the FASTA file for the reference genome sequence, then you can use the VEP to annotate your variants of interest as well. So I mentioned this briefly, but you can use the VEP with the web interface, which is what we're going to look at today. We have a standalone Perl script, and this is really important. I think if you're aiming to do sort of whole genome variant analyses, the web interface is going to be very slow and time-consuming for you to use, so you would be much better off using our Perl script. And there's a wealth of documentation on how to use this online, and you can always email help desk if you're looking to transfer to using that. You don't really need to use understand how to use Perl coding programming language, but we'll help you set that up if you need to do large-scale analyses. And you can also find it through the REST API, which is what Emily's going to speak about next week. Okay, so that summarizes what I want to say quickly about the background information for the VEP. We're going to launch into the demo now. We've identified 4 variants on chromosome 9, so a single deletion and then several SNPs, and we're going to use the Ensembl VEP to find out whether these are already known, what genes are affected, and how to prioritize these variants. So I'll jump out my presentation. I've handily already formatted these correctly in my notes, so I'm just going to copy these, and we're going to go back to our browser. So I'm just going to click on VEP at the top here. Actually, I'm going to click on Tools first, because I want to highlight that this is where you'll find the linkage disequilibrium calculator that I pointed out earlier. So if I click on VEP now, we get to this submission form. So if you're ever stuck on anything, all of these question marks in Ensembl will help open help in a new tab or a new window. So if you need help on anything here, you can always click on that. By default, we've gone to human, which is the most popular species in Ensembl, but you can quickly type and filter and find your species of interest using the search box. I'm going to stay on human. I'm going to name my job webinar, and which will help me find it later if I've run a number of jobs. And I can paste my data in here, but note that you can also upload a file or specify a file URL. You can see here that the file size limit is 50 megabytes. So as I said before, if you're planning on doing any large-scale analyses, you really should try and use our VEP script. So we mentioned these 4 different formats you can submit data in, and you can see that we provide examples of these if you need help in formatting your data correctly. So you can just quickly click on these and see examples of each of these in the VEP themselves. I'm going to just highlight and delete this and paste in my data. So what I've listed here is the Ensembl default format. So I've specified my chromosome number, the start coordinate, and the end coordinate, which, because these are single nucleotide variants, the start and end coordinate is the same. specified this positive sign here, indicating that this is a forward-stranded variant. And I've also given myself I've also given my variants these unique identifiers here. You see that we're presented with this option to run instant VEP, and this, if you click on it, will just bring a pop-up with some summary information to show you what this variant is up to in reference to the human genome sequence. So if I close that now, we'll explore all these different options. So it's compulsory to choose which transcript database you want to use. So by default, it's looking at our database, so our Ensembl transcripts. But see that you can change this to looking at both Ensembl and RefSeq, or RefSeq alone, or the GenCode basic ones, which will be a reduced set. We can see that we have these 3 different options here, which we're going to walk through now. So we can expand this for identifiers and frequency data. So by default, we select gene symbol. Gene symbol is just another word for gene name. The symbol comes from the HGNC consortium, so HGNC symbol just means gene name. You can add on any of these other identifiers. So Emily spoke about the CCDS in her previous presentation. Protein identifiers from UniProt, and also if you wanted to translate your variants into HGVS format, then you can click this option and that will put that in your output. And here we can see that we have our first plugin. So anything reported with this P in brackets is a plugin. And if we come to the bottom of this box, we see that we have a little link here, so you can explore all the different plugins that we have available. And now we come to this frequency data section. So we always want to identify co-located known variants, so we've put this as default. Note that you can also have an option to not compare alleles. So if we leave it just as yes, then we're only going to see variants in the output that have the exact same alleles as the variant that we input. So if we have a particular SNP, particular location, and we report A/C, and the allele that we have in the database is A/G, then by default we won't put that in the output because it has a different impact. But you have the option to include it if you're interested. So by default we just use the 1000 Genomes project, but you can add in this nomad data as well here. And we will show you all the PubMed IDs for co-located known variants. And you can see that we have this option to include the flagged variants. So these are ones that I mentioned in the presentation that haven't quite passed our quality control. And you can include these if you're just interested in finding any variants that overlap with your variant data at all. If we expand this extra options section now, we have a section called miscellaneous, but this is essentially looking at your transcript and the transcript quality mostly. So we'll report the transcript biotype, which I think will be useful for everyone, which tells you if it's protein coding or not, as well as these 2 identifiers, which Emily explained, transcript support level and APRIS, which tells you how essentially how likely this transcript is to be one of the main isoforms for the gene. We can have this option to identify canonical transcripts. So this is something that comes up quite often. We at Ensembl don't support a single canonical transcript for a gene because often transcripts have different roles within different tissues and are equally important under different circumstances. So we try not to identify a single canonical transcript, but you have the option to include that here, and essentially this is just the longest one that we have in the database. You have the option to look upstream and downstream of your variant to find, so it's useful if it's an intergenic variant, you might want to see that it's upstream of a particular gene or downstream of a particular gene. And here we come to our pathogenicity predictions. So we have SIFT and polyphen turned on by default. You also have this option dbNSFP, which is a catalogue essentially of these different types of algorithms. So if you click to enable this, you have a whole range of scores here, which can add value to your output and annotate your variants. So I'm obviously not going to go through all of these right now because there's quite a lot of them. But if you click on here and look at this README, then you can maybe identify ones that will be useful for you. And we have another couple of pathogenicity predictions ones, including a loss of function plugins as well. So we come now to regulatory data. By default, we will show you if your variant overlaps regulatory region, but you can also limit this by cell type, as I mentioned before. So if we click on this, then this will limit your output to only show you regulatory features which are active within certain cell types. So we can click here and turn on killer cells or any of these different cell types that we have available. So this is only going to be available for human and mouse, as this is the only species that we have available regulatory data for. And then there are some more plugins here, which I'm not going to go through too much, but looking at splicing, predictions, and conservation. And finally, we come to filtering options. So this first option is about filtering by frequency. So you may want to exclude common variants if you're looking for particularly ones that you think to be rare. And this will take it down to those which are in less than 1% of the global population based on the 1000 Genomes Project. You can set this to any specification that you'd like though, so you can do it from the combined population within certain populations of these large populations, so not the subpopulation, so you can't look particularly just at Britain, for example. You have to look at the whole European population, and you can change these as you see fit. I'm going to leave this as no filtering though. You can return results for variants encoding exons only, and you can even restrict your results. So if you're running quite a lot, you might want to only see one consequence per variant, just summarize it to one consequence per gene, for example, and you can do that here. But as it notes, you may exclude biologically important data, but if you're wanting to filter down your list, you may like to do that to begin with. And now if I click run here, we can see that my options have disappeared, but I've come up with this little table, and it's telling me that my task is queued, and these options at the side here, we have options to save it. So if you've created an Ensembl account, you can save your queries, otherwise they'll just hang around until you refresh your browser and delete your cookies. You have options to quickly edit and resubmit the ticket, and importantly to share your ticket. So if you've inputted certain variants and you've turned on and off all different kinds of configurations in the submission box, which we looked up just a moment ago, then you can share this with your colleagues and they will see your output and all of the options that you've put in to filter the data set as well. So that's very handy if you need to share this with your colleagues, and then quickly you can delete the ticket if you're interested. So you can see that this is refreshed now. This refresh button at the top was counting down, and now it says done, and I can just click on view results. So at the top of the page we have this table which tells us how many variants were processed, so 4, so that's good because I put 4 in, and if any were rejected they'd appear in the second row. And we have a ratio of novel to existing variants, so you can see that 1 of mine, or 25% of my variants, were novel. We can see that this overlaps 2 genes and 1 regulatory feature. So this first pie chart here is showing us the consequences of across all transcripts for all of my variants. And the second one is just looking at a subset of this, so looking at those which overlap coding regions and what the consequences of those are. So you can see the majority is missense variant, but we have a synonymous variant as well. So this is one that has changed the base sequence, but not the amino acid sequence there. So if we come down to this main table, this is showing us all of our results. So you can see my uploaded variants, they're not in the same order that I submitted them in. So that's why it's sometimes useful to provide these IDs, because it's otherwise going to default order it by location. We can see what the allele is, the alternate allele that is. So this is our deletion down here. And if we scroll across, we can start to see our consequences, what the symbol is, so what genes are affected, so COC4 and TRUB2, as well as the identifiers, which if we click on will take us directly to the gene tab and the transcript tab respectively. You see that we have one here that's overlapping a regulatory feature, and we know this to be a promoter, and we also have a link to look at the stable ID for that promoter. If we continue to scroll across, we can see we have this column existing variant. So if we scroll down, we can see that that deletion, my variant 1, is the one that is not identified already. And you can see that we have SIFT and polyphen scores available for those which were identified as missense variants. So you can see this one is not particularly damaging, but this one here is predicted to be quite significant. So that is overlapping our COC4 gene and is our third variant here. So this is quite a large table. You can see that we've come to the minor allele frequencies here. And you may want to filter your table just to find things of interest to you. So for all of these columns in the table, you can filter. So if we want to filter by consequence, for example, we can start typing in missense, and it will filter the list based on all of the categories available in that list. And if I click Add, you can see that my table is reduced now. Just to look at variant 3 and 4, which are the only ones which had missense variants, and you can see that this has appeared at the top here. What we can do is also apply another feature, and that's filter, and that will also decrease the list. So I can filter by SIFT and say I want it to be less than 0.1, for example, and add that as a filter. And now you can see it's reduced even further, and now I'm only looking at my very deleterious or annotations for the SIFT. And so you can apply as many filters as you want, and once you've got your table looking how you want it, you can then download it. So you can download it as a VCF, VEP, or a text document, and you can also export your results to Biomart for our variant dataset or gene. So once you've identified all of the genes that that are affected by your variants, you might want to find out more information about those genes, such as their phenotype. So you can just quickly click on this, and it's going to export all of your genes from this table into Biomart, and you can download a wealth of data about that, which we're going to take a look at next week in our Biomart section. And the same is true for variants. So wherever you've identified known variants, you can also export these to Biomart and download a wealth of data about these. Okay, so I'm going to go back into my presentation now and just wrap up. As I said, all the course exercises are located here, so at this link, and you'll be able to find the variation section where this video will appear online, and also links to the exercises and then also the solutions, so you can work through those in your own time. And if you need help, as I said, please post on our Slack workspace, and also just email us at help desk if you've got any particular questions that you would not like to post to the Slack channel but want to ask us directly, then you can put those in here, and they can be related to anything to do with Ensembl, not just these webinars and the exercises. And so leading on from this, we're going to have comparing genes and genomes with Ensembl Compara, which is going to be led by Astrid, so we'll start that in about 5 minutes time. And this is just a summary of what this is going to cover. So we're going to be looking at how you can detailed analysis of gene models between species, so looking at gene trees and orthology and pyrology, but also at whole genome alignments between pairs and groups of species. Okay, so thanks for coming along, and stick around to see Astrid's talk.